Dear students, today we will study Herbarium. I am Rajkumar sir. On the right hand side of the slide, you see the specimen of Adonis vernalis Linn. This was collected by Ulisse Elrovendi in 1561. This is one of the oldest herbarium specimens. You see, so now what is an herbarium? Herbarium क्या होता है? Herbarium is a collection of pressed and dried plant specimens which are mounted on sheets which bear a label, and these sheets they are arranged according to a particular sequence and they are available for reference or future studies. However, in practice, this name is given to a place which is owned by an institution that maintains this orderly collection of the plant specimens. This gentleman, this scientist is Luca Ghini, and he is credited for the first recorded herbarium as well as for the first botanical garden in the Europe. He was an Italian physician and botanist, although none of his herbaria survives till today, but those created by his students for Gerardo Cibo 1512 to 1600 and Ulisse Elrovendi 1522 to 1605 still survives. This art was disseminated throughout the Europe by his students who mounted the sheets and bound them into book volumes. When Linnaeus started his experiments, Linnaeus, instead of mounting and binding the specimens into volumes, mounted the specimen on single sheets and then stored them horizontally, and this practice is followed throughout the world even today. From isolated personal collections, the herbaria have grown into large institutions of national and international stature with millions of specimens from the different parts of the world. In India, Central National Herbarium is maintained by Botanical Survey of India. It is located at Havda. On the right hand side, you see a picture of oldest herbarium specimens in India. This was collected in the year 1696 in the month of June by Dr. Samuel Brown from a locality between Madras and Tirupati. This is a specimen of Cyprus procerus rotab. Index Herborarium Riorum, which is edited by Patricia Holmgren, provides a global directory of herbaria and their associated staff. This is a searchable online index and it allows scientists to for the rapid access to data which is related to 3400 locations where a total of 350 million botanical specimens are permanently housed. Over a period of time, six editions of the index were published from 1952 to 1974. The index became available online in 1997. Now we will study some major herbaria of the world. Each herbarium is identified by an abbreviation, and this abbreviation is valuable in locating the type specimens of various species. We will study about 10 herbarias. Museum of Natural History is located in Paris, France. It is abbreviated by P or PC and it houses 10 million specimens. New York Botanical Garden, New York, USA is abbreviated by NY and it contains 7.8 million specimens. Komarov Botanic Institute, St. Petersburg, formerly it was known as Leningrad. It is present in USA and it is abbreviated as LE and it houses 7,180,000 specimens. Royal Botanic Gardens, Kew, Surrey in UK is abbreviated by K and it has 7 million specimens. Missouri Botanical Garden, St. Louis, Missouri, USA is abbreviated by MO, and it contains 7 million 6, 6.6 million specimens. Conservatory and Botanical Garden at Geneva, Switzerland has an abbreviation of G, and it contains 6 million specimens. British Museum of Natural History in London, UK is abbreviated by BM. It has 5.2 million specimens. Combined Herbaria at Harvard University, Cambridge, Massachusetts, USA is abbreviated by AFHGHECONAMES. The number of specimens present here are 5 million. Natural History Museum, Vienna in Australia is abbreviated by W, and it contains 5 million specimens. US National History Herbarium, Smithsonian, Washington, USA is abbreviated by USA, and it contains 5 million specimens. In India, Central National Herbarium is abbreviated by CAL since it is located at Calcutta. Indian Botanical Garden, Botanical Survey of India, Kolkata has over 2 million specimens. The Herbarium of Forest Research Institute, located at Dehradun, Uttarakhand, is abbreviated by DD. National Botanical Garden, sorry, National Botanical Research Institute, Lucknow, is abbreviated as LUCK. Are other major herbarias in India with collections from all over the world. Now, how a specimen is prepared. A specimen meant for incorporation in herbarium needs to be carefully collected, pressed, dried, mounted, and finally properly labeled so that it can meet the demands of rigorous taxonomic activity. जो स्पेसिमन हर्बेरियम के अंदर रखना है उसको बड़े ध्यान से कलेक्ट करना है, डिजीज़ फ्री कंडीशंस में कलेक्ट करना है, उसको प्रॉपरली प्रेस करना है, ड्राई करना है, उसके बाद शीट्स के ऊपर माउंट करना है और ढंग से सही तरीके से लेबल करना है ताकि जो है भविष्य की टैक्सोनॉमिक एक्टिविटीज़ जो भी उस स्पेसिमेन के ऊपर करनी है ढंग से की जा सके. Specimens are properly prepared so that they should retain their essential features for very long periods. This proves to be immensely useful for future scientific studies, including compilation of floras, taxonomic monographs, and in some cases, even experimental studies. Since the seeds of several species can remain viable for many years, इवन इन ड्राइड हर्बेरियम स्पेसिमेंस, क्योंकि बहुत सालों तक सीड्स जो हैं ड्राइड कंडीशंस में वायबल रहते हैं, जिंदा रहते हैं, इसीलिए इन्हीं हर्बेरियम शीट्स से ये सीड्स निकाल कर बहुत सारी एक्सपेरिमेंटल स्टडीज़ भी की जा सकती हैं. फील्ड वर्क. The field work of specimen preparation involves plant collection, pressing, and partial drying of the specimens. The plants are collected for various purposes, and the trips or the field work activity are of three types. One is collection trip, which is of short duration, usually 1 or 2 days, to a nearby place for brief training in field work, vegetation study, and plant collection by group of students. Next, exploration. This includes repeated visits to an area in different seasons for a period of few years for intensive collection and study. It is aimed at compilation of floristic accounts. The third type of field work is expedition. Such a visit is undertaken to remote and difficult area to study the flora and fauna, and this usually takes several months. The equipments which are necessary for field work are The plant press, field notebook, bags, vasculum. Instead of vasculum, nowadays polythene bags are used. Pencil, cutter, pruning shears, knife, and digging tool. Now, first the plant press. Plant press consists of two wooden, plywood, or wire mesh planks, and each is of dimension 12 inches into 18 inches, between which are placed corrugated sheets, blotters, and newspaper sheets for the proper drying of the plant specimens. Two chains or belts are used to tighten the press. Corrugated sheets or ventilators are made of cardboard and helps ventilation and the consequent drying of the specimens. Newspapers are changed frequently when they absorb sufficient moisture. Field book. A field notebook or field diary is a very important item. It contains numbered sheets with printing pro forma for entering field notes such as the scientific name, the family name, the vernacular name, locality from where the plant specimen is collected, the altitude of the locality, the place, the date of collection, and for recording any additional data that are collected in the field. There are multiple detachable tags with serial number at the lower end of the sheets, and these tags are used for multiple specimens of the species collected from a site. It serves as ready reference to the information recorded in the field notebook. The number is the collection number for the collector. This is a sample of the field notebook page where you can see that there is plenty of information which has to be recorded. Next, the vasculum. Vasculum is a white colored metallic box with tightly fitted lid and a shoulder sling. This is used to store specimens temporarily before pressing and also to store bulky parts and fruits. It being heavy, it is nowadays substituted by polythene bags, which is almost weightless, and they are readily made airtight using rubber bands. While collection, it should be noted that the specimen collected should be as complete as possible. For example, herbs, very small shrubs, should be collected complete in flowering condition along with leaves and roots. Trees and shrubs should be collected with both vegetative and flowering stages. All information concerning the plant should be recorded in the field notebook, and a tag from the sheet is attached to the concerned specimen. Always collect a few specimens of each species from the site to ensure that reserve specimens are available if one or more get destroyed, and also to ensure that duplicates can be deposited in different herbaria when finally mounted on sheets. If, if for example, say you have to send your herbarium specimens to some herbaria do ask you to permanently deposit your specimens. They do not return your specimens. They keep their specimens for their records after identification. So you must have a duplicate copy of it. Now, pressing of the specimens. A specimen shorter than 15 inches should be kept directly in the folded newspaper after loosely spreading the leaves and branches. Herbs or shrubs which are longer than 15 inches, they should be folded in the form of V, N, or W. The terminal part of the plants with leaves, flowers, and fruits, it should be in the erect position. The specimens of Grasses and some other groups which show considerable elasticity are difficult to hold in folded condition. These specimens can be managed by using flaxostats. Flaxostat is a strip of stiff paper or card having 2.5 centimeter long slit, and they are placed at each corner of the specimen. This holds the specimen firmly. To press bulky fruits, the fruits can be thinly sliced. Large leaves can be trimmed to retain any lateral half. It is useful to invert some leaves so that the undersurface of the leaf can also be studied from a pressed leaf. This is how the folding of longer herbarium plants is done. Now, coming to the drying process, the drying can be done in two ways, natural and artificial. Now, first, natural drying. The plants freshly collected are placed in folded papers in press without corrugated sheets, and the press is locked for 24 hours. This is a sweating period, and during which the plant lose some moisture and become flaccid. And during this period, the plants can be easily rearranged. If they are not properly pressed, one can rearrange them and then again press them. The specimen should be carefully and delicately placed in a fresh set of dry folded blotter or newspapers. The change of blotters or newspaper sheets is repeated every few days in increasing the interval between the changes successively until the specimens are fully dry. The whole process of drying by natural means may take place about 10 days to 1 month. This depends upon the specimens and the climatic conditions of the area where the drying process is conducted. One can dry using artificial heat also. The drying with the help of artificial heat takes about 12 hours to 2 days. This considerably shortens the drying period. The specimens, after the initial sweating period in the field press, they are transferred to a drying press with and corrugated sheets. Usually, one alternating every folded blotter containing one specimen. The press is kept in a dryer, a cabinet in which a kerosene lamp or electric bulb warms the air, or the use of hot air blower for drying the specimens by movement through the corrugates for faster drying. Synod in 1983 developed a solar powered dryer which was capable of drying 100 specimens on a sunny day. There are limitations of artificial heat, which are first the rapid drying method. This is a rapid drying method with artificial heat renders the plant brittle. There is a loss of bloom and some color change in the leaves. In arid regions, the plants can be dried partially during the travel by placing the press horizontally on the luggage rack of the vehicle with the corrugate ducts facing front, forcing the dry wind through the corrugates as the vehicle moves forward. Pressed and dried specimens are mounted on herbarium sheets with proper labels for incorporation in the herbarium. The mount pressed and dried specimens are mounted on herbarium sheets. The standard size is 29 by 41.5 centimeters, 11.5 by 16.5 inches, made up of thick handmade paper or card sheets. It is relatively stiff to prevent damage during handling of the specimens. It should have a high rag content, preferably 100%, with fibers running lengthwise. The specimens, how the specimens are attached, now we will study this. The In case of older specimens, we see that they have been sewn and Nowadays, or by soon or by using adhesive linens, paper or cellophane strips, they are fixed on the herbarium sheets. The use of adhesive linen, paper, or cellophane strips is an easier and faster method of fixing specimens to the herbarium sheet. The specimens can also be pasted or pasted on the herbarium sheets using glues in two ways. The paste or glue is applied on to the backside of the specimen, which is then later pressed on the mounting sheet. The second method is that paste or glue is smeared on a glass or plastic sheet. The specimen placed is then placed on the sheet and the glued specimen is transferred to a mounting sheet. Small paper envelopes, which are called as fragment packets, are often attached to the herbarium sheet to hold seeds, extra flowers, or loose plant parts. This is a sample herbarium sheet with a label, and this is of Herbarium rosa sinensis, Malvaceae family. Now, what, how to label and now we will study the labeling and herbarium label. It contains information which is recorded in the field diary or the field notebook at the time of collection or results of any subsequent identification process. It is basically a pre-printed proforma printed directly on the sheet or on the paper slips which are pasted on sheets at the lower right corner. The size is 2 3 by 4 inches by 4 1 by 4 inches, or by Woodland 1991, the size is 4 by 5 4 by 6 inches. It contains information like name of the institution, scientific name, common name, family, locality, latitudes, longitudes, altitude, date of collection, collection number, name of the collector, and habit and habitat, including certain field notes. Now, the annotation or determination label. It may happen that an expert visiting a herbarium may want to correct an identification or a record a name change. Such correction is done on small annotation labels or determination labels, which is of size 2 by 11 centimeters, and this is attached left of the original labels. It contains correction, name of the person, and the date on which the change was recorded. More than one annotation labels can be attached. The last label is likely to be the correct one. Now, how the specimens are filed? Mounted, labeled, and treated specimens are finally incorporated in a herbarium where they are properly stored and looked after. Small herbarias, they arrange specimens according to the family, genus, and species in an alphabetical manner. Large herbaria, they follow a particular system of classification. Most of the herbaria usually follow Bantam Hooker. British herbarias and most Commonwealth countries follow Bantam and Hooker. In Europe and North America, Angler and Pruntal classification system is followed for arranging the herbarium specimens. Many herbaria of the later category follow the number code of the families and genera which was given by Dallatore and Harms. The specimen belonging to a species are placed in a folder made up of thin strong paper which is called as species cover. The species cover belonging to a particular genus are often arranged alphabetically and are placed inside a genus cover, which is a heavy manila folder made up of thicker paper. Unknown specimens are kept in separate folders which are marked as dubia, placed towards the end of a genus when the genus is identified, or a family when the family is identified but not the genus, as the case may be, so that an expert can examine them conveniently. Standard herbarium cases are insect and dust proof with two or more tiers of pigeonholes, each 19 inches deep, 13 inches wide, and 8 inches high. Type specimens are usually kept separately in distinct folders or often in separate herbarium cases, sometimes even separate rooms for better care and safety. A herbarium commonly maintains an index register in which all the genera in the herbaria are listed alphabetically, and against each genus is indicated family number and the genus number. These two help in convenient incorporation and retrieval of the specimens in a herbarium. You see, herbarium specimens are liable to be infected by pest controls. Then, how the pests are controlled? Herbarium specimens are usually sufficiently dried, and as such they are not attacked by bacteria or fungi, but they may be attacked by pests like silverfish, dermastid, beetles like cigarette beetle, drugstore beetles, and black carpet beetles. Control measures include treating incoming specimens by three ways: heating at temperatures up to 60 degrees Celsius for 4 to 8 hours, by using deep freezers, which has now replaced heating cabinets in most herbaria of the world. A temperature of minus 20 to minus 60 degrees Celsius is maintained in most herbaria. Microwave ovens have been used by some herbaria, but it has some serious shortcomings, like stem and embryo damage due to sudden vaporization of the water which is present inside the tissues. There is use of repellents like naphthalene balls and para-chlorobenzene, and these are usually powdered and put in small muslin bags kept in the pigeonholes. By fumigation methods, fumigation involves exposing specimens to the vapors of certain volatile toxic substances. For example, a mixture of ethylene dichloride 3 parts and carbon tetrachloride 1 part. Nowadays, there is a concept of virtual herbarium. Virtual herbarium is a database consisting of images of herbarium specimens and the supporting text available over the internet. And it is a huge advancement in herbarium use and design, coupling physical specimens directly with internet and integrating complete specimen data with resources or information generation and retrieval. There are advantages of virtual herbarium. The images being available electronically, so they cause less damage to the specimens. Sorting out of specimens of a collector or a country in a physical herbarium takes very too much time. This same can be done in few seconds in a virtual herbarium. Thousands of users can access the virtual herbarium sitting in the comfort of their houses or offices. Provides information on descriptive details, geographical distribution, photographs, illustrations, manuscripts, published work, microscopic preparations, gene sequences, and nomenclature through hyperlinks. The portals of major virtual herbaria offer facility of searching several virtual herbaria simultaneously. Thousands of specimens and valuable holotypes preserved in Berlin Herbarium were destroyed during the Second World War. The digitalized images, on the other hand, can be saved on several computers at different locations, and so that they can be there is no loss to it. Majority of the herbarium projects do not need physical specimens, and as such electronic images can be utilized. This saves the time and cost for transportation of actual specimens. Virtual herbaria with searchable databases have been developed by major organizations like New York Botanical Garden, Royal Botanical Garden, Melbourne, Fairchild Tropical Garden, Australian Virtual Herbarium, Royal Botanical Garden, Kew. Now we will study the functions of herbarium. First, it acts as a repository of plant specimens. The primary role of herbarium is to store dried plant specimens and safeguard these against loss and destruction by insects and make them available for study. The second important function is the safe custody of type specimens. The type specimens are principal proof of the existence of a species or an infraspecific taxon. These are kept in safe custody, often in rooms with restricted access in major herbarias. Compilation of floras, manuals, and monographs are largely based on herbarium resources. The herbarium offers training in herbarium methods since the herbarium herbaria people are expert in this field. They offer facilities for graduates and undergraduates. They organize field trips and even expeditions to remote areas to train these people. They offer facilities for identification of the specimens. They also provide information on the geographical distribution of a taxon. They serve for the preservation of voucher specimens. Voucher specimens are preserved in herbaria, provide an index of specimens on which a chromosomal, phytochemical, ultrastructural, micromorphological, or any specialized study has been undertaken. In the case of a contradictory or doubtful report, the voucher specimens can be critically examined in order to arrive at a more satisfactory conclusion. The classification of the world's flora is based mainly on the herbarium materials. The plant specimens are permanently stored in herbaria and therefore they are the major sources of information about plants and vegetation. Herbaria preserves the national plant wealth and provides scientific information to the public regarding the plants. There are several identification methods. Identification of an unknown specimen and determination of the correct name. The combined activities appropriately referred to as the specimen determination. Before the specimen can be identified, it is desirable to describe it and prepare a list of characteristics mainly pertaining to the floral structure. Whereas fresh specimens may be described more conveniently, the dried specimens may be softened by immersing in water or a wetting agent such as aerosol OT, which is sodium bis 2-ethyl hexyl sulfosuccinate 1%, distilled water 74%, methanol 25%. Pohl's softening agent is an excellent detergent solution for softening flowers and fruits for dissection. The identification of an unknown plant may be achieved by comparison with the identified herbarium specimens or through the help of taxonomic literature. The unknown specimen meant for identification is sent to a herbarium where an expert on the plant group examines and identifies it by comparison with the duly identified specimens. The user can also visit a herbarium and personally compare and identify his specimens. The photographs, description, or illustration of parts can be put up on a website with information to a relevant email list whose members can help in achieving identification within hours. Now there are precautions for using herbarium. First, since the plant specimens are brittle and they're easily liable to be damaged, so the Specimen sheets should be kept flat and they should be handled with extreme care because they are of very important. They carry very important scientific values and they are generally irreplaceable. Always store the specimen in herbarium cases. Keep the materials in folders when not in use. Never keep heavy books or other objects on the specimens. The support the specimens with a ventilator when carrying them even for short distances. If you notice that the specimens are being damaged by insects or other pests, one should inform the curator immediately. One should not write anything on the sheets of herbarium unless he or she is permitted to do so. Do not reshelve specimens in herbarium cases. If you have taken specimens on loan basis from any other institution, one should return them before the loan period is over. The specimen borrowed from other institutions should be properly housed in fireproof, dustproof, and pest-free cases. Now, we can answer few questions by studying the lecture. What do you mean by the word herbarium? Name the largest herbarium of the world. Is it located and in which country? Write a brief note on modern herbarium. Make a list of 5 major herbaria of the world and also 5 major Indian herbaria. Write 6 general functions of herbaria. Comment on modern herbaria work as service institutions. What precautions does a herbarium require? Now there are certain references. One can see Plant Taxonomy by O.P. Sharma 2017, Gurucharan Singh's 2019 Plant Systematics and Integral Approach of 4th edition or 3rd edition, M.G. Simpson 2006 Plant Systematics. And thank you for watching it. Revise it, learn it and subscribe the channel. Thank you very much.